Hello, my name is Alfredo Garcia. I'm a student at Grand Canyon University, and today I'm going to be presenting uh, a discussion on a specific article titled Silver Nanoparticles, a Novel Antibacterial Agent for Control of Chronobacter sakazaki. So, a little bit of an introduction. Silver, which I believe everybody knows what it is a soft, shiny metal, usually used for making jewelry and other trinkets. We've known about it since ancient times. It's been used medicinally for over 600 years. And before antibiotics, it was actually considered to be the most important antimicrobial agent. So, humans, have known about the antimicrobial properties of silver for many, many centuries. And it was actually used to keep water and food fresh for longer. So, with the advent of antibiotics, however, silver fell sort of out of favor as an antimicrobial medication, but recently, silver nanoparticles have raised interest as a possible antimicrobial agent once again. So, what has changed? Nanoparticles don't behave like their bulk counterparts and have different properties. So, a little bit of background: nanoparticle is described as being less than 100 nanometers, and these particles behave much more differently than bulk counterparts. So they, for example, ionize much more easily, interact with other particles better because of their increased surface area, and may even be a different color or have different physical properties such as melting point. So because these silver nanoparticles have such different properties than their bulk counterparts, silver is again being studied as a possible antimicrobial agent. So recently, 2018, Wang et al., so Wang and others, published an article in which silver nanoparticles were explored as an antimicrobial agent against a specific microorganism called Chronobacter sakazaki. So, what is Chronobacter sakazaki and why should we kill it? Chronobacter sakazaki is an infectious agent, in specific a bacterium, that infects the bloodstream and nervous system, which makes it usually fatal. So it's usually ingested and acquired as a source of food poisoning. So it's a very dangerous disease, especially for newborns and babies born with low birth weights. So the means that the babies usually get these infections is from powdered baby formula. So for this reason, Wang et al. Wanted to explore silver nanoparticles as a possible means of killing the organism in the production of baby formula. So their methods, they performed a study as a primary test of antimicrobial properties of silver nanoparticles against Chronobacter sakazaki. And the reason for this is that they want to determine this before the material is implemented. So you can't implement a material before you know if it's going to be effective at killing the organism which you're trying to eliminate. So the silver nanoparticles were derived from silver nitrate using the methods described by Day et al. In 2015, and 4 strains of Chronobacter sakazaki were tested. 2 types of assays were performed in order to test the efficiency of the silver nanoparticles as an antimicrobial agent against the organism, and these were the minimum inhibitory concentration test and the inhibition zone test. So I'm going to explain briefly what these 2 tests are in a moment. So the MIC test, or minimum inhibitory concentration test, is a simple assay for testing the inhibitory properties of a chemical against a specific organism. So you get a sterile tube containing clear liquid growth medium and you just inoculate it with the organism and incubate it. So if whatever you're testing is good at killing the specific organism, you shouldn't see any growth. If it's not effective at killing the organism, then the liquid growth medium is going to get cloudy and you should be able to visually see the growth of the organism. So they performed this test using 10 milliliters of Luria Bartani liquid medium against 1 milliliter of the 4 strains of Chronobacter sakazaki. Silver nanoparticles were added to the growth medium, and the positive controls were test tubes with only the microorganisms added, and the negative controls were test tubes with only growth medium present. The test tubes were incubated at 37 degrees Celsius for 12 hours, and the organism survival was then inspected visually. So the IZ test, or inhibition zone test, is also an assay, but it's performed on a solid growth medium instead of a liquid growth medium. So a petri dish. The zone of inhibition test, a sample of about 1 million cells is placed on the growth medium, and the antimicrobial object is usually placed in the center, or depending on how it's done, sometimes you get several types of antimicrobial objects put in there, which was the case with the study. But if the object or chemical is inhibitory in nature, then you should see an inhibition zone form in the dish, and you can actually see that in the picture what it looks like. So Wang et al. Performed this test using the Oxford cup method and a double-layered growth medium, which consisted of water agar and a nutrient agar layer. So about 100 microliters of each strain was split on the plate, followed by another 100 microliters of the silver suspension were added at different concentrations to the whole, and the Oxford cups were removed. So Oxford cups are little cups that you place inside the growth medium and then just put whatever you're trying to put in there and then just yank them out, and whatever you're trying to get in there stays in there. So the positive controls were a hole with silver nitrate, and the negative control was a hole with sterile water. They incubated for 12 hours at 37 degrees Celsius also, and after the fact, the inhibition zones were examined visually, and the diameter of the zone was measured to get an account of the level of inhibition. So the results of this study, the Mike test results showed that silver nanoparticles did show antimicrobial properties which were strain specific to the 4 strains of the organism. The IC test was also positive, and the nanoparticles were observed to form an aseptic area and stronger antibacterial properties than regular silver ions on all 4 strains of the Cronobacter sasaki organism. The bacteria were also tested after the fact to obtain a measure of cell membrane integrity, membrane potential, leakage of sugars and proteins, and intracellular ATP concentration. So the reason for this was to sort of try to figure out after the fact not just if silver nanoparticles would kill the organism, but how they were going about killing the organism. So what they found was that the silver nanoparticles reduced cell membrane integrity, hyperpolarized the cell membrane, increased protein and sugar leakage, and reduced intracellular levels of ATP. So this all coincides with the observation that silver nanoparticles are antimicrobial in nature against the 4 strains of Cronobacter sakazaki. So how do we tie this into current events? So this article ties into this, our current event course in 2 ways. It connects with medicinal chemistry and materials chemistry. So I believe that this is more so related to medicinal chemistry, as it is something that is being tested in order to try to treat a specific disease. But I myself found the materials component more exciting. So this is because the metal, which has been known since ancient times, so we've known about silver since ancient times, and we thought it had lost all medicinal value in modern times. So antibiotics just completely replaced it, and now we're re-looking at this metal which we've known about for such a long time and somewhat repurposing it based on these different properties that it exhibits as a nanoparticle. So as I stated before, nanoparticles of a given material can exhibit different chemical and physical properties than their bulk counterparts. And this means that there could be many elements and compounds which may perhaps be repurposed and find new interesting uses based on these changed properties. So gold nanoparticles, for example, are also very different than bulk gold. Gold is oftentimes referred to as a noble metal, meaning that it doesn't really react with other chemicals. And it's also not very magnetic, but as a nanoparticle, it becomes reactive and magnetic, which means that gold nanoparticles could be repurposed and used for completely different things than their regular gold counterparts. My conclusion, so I believe that Wang et al., that the study was very well performed, as the initial investigation into the possible uses of silver nanoparticles as a means of fighting Cronobacter sakazaki. So they performed 2 separate tests to identify if the silver particles were antibacterial in nature, which is good. So you don't you want to try it out a bunch of different ways and see if you get the same results. They utilized 4 separate strains, which is important in with bioorganisms, because oftentimes it's possible for separate strains to have different levels of resistance to certain chemicals. So the only concern that I can express against the article is not utilizing silver directly as a means of treating powdered baby formula may not be possible. So using silver nanoparticles to treat baby formula may not be possible. And that's because silver, while not toxic, does build up in tissues and causes a condition called argyria, which results in a great discoloration of the eye, skin, and organs. And I don't really think that anybody wants to expose their babies to that. So maybe silver nanoparticles could be applied indirectly. So that's something that would need to be studied in the future. What the effects on people and infants are. And so what may be done perhaps is that the nanoparticles maybe can be applied directly to the machinery used in making the formula, or maybe to the inside of the container, to the material that the container is made out of, as a means of keeping the baby powder sterile while it's packaged. As for approaching this from a Christian worldview, I do believe this is a very noble undertaking because it's aimed at helping some of the most vulnerable members of our society, which are babies, neonates, and specifically premature or underdeveloped babies. So how does this affect us? The current repurposing of materials affects us greatly both as chemists and as people. So as chemists, it opens up a new field of research in which materials which we thought we knew almost everything about are re-explored. As just people, it may lead to advances in medicinal chemistry which may make treatment of prevention of specific diseases much easier. So the paper by et al. In specific affects us greatly because it perhaps offers a means of preventing a deadly disease which affects some of society's most vulnerable members. So, like I said before, the most exciting part about this, I think, is just knowing that there are so many materials out there, both compounds and elements, which maybe we can find new uses for based off the different properties that they exhibit as nanoparticles. And learning how to treat infectious diseases is something which will always benefit humans because we're always trying to find new and better treatments for diseases. So I'd like to thank all of my citations, which I've included, and I want to thank everybody for your time. So whoever hears this, thank you very much.